So we have about a half hour left before we end this call, and I wanted to sort of broaden our talk beyond CROI a little bit. And I really wanted to ask our four guests, what should we be paying attention to in terms of HIV cure in the coming years? So, like, let's give it a 5-year span. What do you think we should be focusing on as community members when it comes to HIV cure? What should we keep our ears peeled for, so to speak? So why don't we start with random selection, Anne?
Um, well, I think it's we're poised to have a lot of excitement come out in the next 5 years. I mean, I think everybody on this call knows that there are so the recompetition for the Martin Delaney collaboratories. So those will be reviewed very soon. And one very exciting thing, I don't know if everybody is aware, but there was a specific call for a pediatric focused Martin Delaney collaboratory this year. Yes.
Mhm.
And so hope I like all the celebrations. So, regardless of who gets that, there will be a lot of exciting research that comes out from that. And I think, just all of the efforts to apply the cutting edge techniques that are being developed to understanding the reservoir in perinatally infected children and adolescents is really, it's going to be very exciting to see where that takes us. And then, of course, where my heart is to really try to push the envelope in the infant non-human primates to try a bunch of different things and different approaches and see if we get any signals of efficacy and obviously safety, and then to kind of move forward with those in human clinical trials. I think, many people who are involved in planning clinical trials in adults, I've heard again and again that there's not really a you don't need to have a non-human primate safety study first before moving forward, depending on what the product is, obviously. But in pediatrics, there's much more reticence to new novel interventions that haven't been tried first, either in adults or in non-human primates. And so that's what we're really trying to do, is test, things that we can do when ART is initiated, because we have kind of more ready access to infants soon after birth, so we can add on things in addition to very early ART, but also not lose sight of all of the perinatally infected, school age, adolescent, and even young adults that are out there right now living with HIV that also demand a cure too. So we're in at least in our approach would be to try to focus on all of those different groups.
Thank you for that. There's a related question posted in the chat after you all make your way through responses to Michael's question.
Sure. So why don't we have Luis?
So, I mean, I think in the most immediate future, the thing to look forward to is the rebound of all the trials that were affected by COVID, and that we will then move forward. I think there was a lot of the research activity that got hit last year that is hoping to recover and move forward this year. So I think we're going to see a lot more trial outcomes next year than we did this year in the short term. I think from a scientific point of view, as we talked about earlier, this whole idea of how best to be measuring strategies by what they're doing to the reservoir and taking advantage of these new predictors for rebound would be the two tools that are in our kits now that moving forward would be of interest to use and exploit. But I think from an activist point of view, I think, we'll take care of the science. What you need to take care of is that NIH doesn't lose the eye on the ball. And I believe that, they're now talking about getting rid of the UO1s as trial independence of the ACTG, and how there may be merging trial structures into the ACTG to try to come up with hybrid, types of settings, which is great because we talked about that before, right? How the ACTG should reach out more to outside people from the ACTG, but securing that they create a structure that protects resources that could be accessed by the outside folks I think it's going to make sure that we have a very diverse effort nationally. And so I think that's one area that I would encourage you to learn more about. And the letter for the fetal ban lifting was sent, so we're all waiting to see whether the Biden administration is going to do something about that. But I think that from a cure perspective, that was really an important step. And following up on that as to what's going on would be useful for the research effort. And then I've also talked about before centralizing some of these high cost technologies, like to make them more accessible to research programs like whole genome sequencing, KIR, and HLA typing. There's a series of very core types of resources that are highly specialized, very costly, and would take huge chunks out of our grants if we put them in. But we would be so much better if we had access to them. And so pushing those for greater access would be great.
Thank you. And Professor Jones.
Yeah, so there's three areas I would highlight. On the clinical trials front, there's a lot of activities that, as Lewis nicely put it, are going to be taken off the ice and moving forwards. There's several studies that combine broadly neutralizing antibodies with immunomodulatory agents, be they TLR7 agonists or IL-15 superagonists, that I think we should keep an eye on. I think there's reason to be cautiously optimistic on the basis of results from non-human primate studies with these same combinations where some of the animals have gone into long-term remission. On the other hand, as Lewis also pointed out, there was a poster from CROI with treatment with 2B NAbs in early infection that differed from the previous results with other antibodies from Malcolm Martin. So that's a bit of a note of caution, but there are several reasonably sized studies moving forward in people soon. And I'm going to be keeping a close eye on those for sure. In terms of the more basic science, I really want to know what's going on with this active reservoir that is getting a lot more attention from a number of different fronts. So I think we know now that the reservoir doesn't simply hide from the immune system. It's more complicated than that. And any clues that we can learn regarding how or why that's happening, I think is going to open up new therapeutic fronts. And then the third thing I would highlight is that I really hope in the next few years, we see a lot more activity towards working with non-subtype B virus and reservoirs and looking at cure from a more globally diverse perspective. So a lot of the new assays that have been developed, in particular, I would highlight the IPDA, have the potential to be more scalable and usable in less resource intensive ways. But there's work to be done to make those assays work with the types of virus that are circulating in places other than North America and Europe. And I think that by extending those studies, not only will we set the stage for more equitable interventions when they come around, but also I think there's going to be differences in the way the reservoir behaves in different populations and different subtypes. And having those diverse perspectives might lead us to some fundamental insights about how HIV persists in a really general sense.
Right.
Yeah.
Thank you, Brad. A couple questions in the chat, but before we get to the questions, Linda D, really quick, your question.
I wanted to say to Luis that I'm sure the fetal tissue stuff that they'll let us know as soon as they hear something, the organizers of that letter. And I think that what they're doing with the ACTG is to allow for the stuff that we talked about. I think I talked to Carl about that. Allow for some more money for the needs of, for instance, the MDC working with the ACTG. But I wanted to know you said UA ones, right? Not our ones. I mean, they're still, right?
Yeah.
What did you say, Luis, about what's going to disappear?
there's been, in the trial development platforms that are available through grants, the UO1 is called a cooperative agreement, meaning that
Right.
and it's the same magnitude of funding as an R1, except that an R1 is you have a principal investigator driving the research.
Right.
In a UO1, there is more involvement from NIH program in the team, like a trial would require. So that if you have a trial concept, then you would submit a UO1 in order to get it done. Now, the question is, when it comes to the cure agenda, are they going to say you can only do them in the ACTG through this new structure? Or are they going to allow both of these to coexist? I would argue that until they show that the ACTG is not going to be a closed shop in spite of how they design it, that the UO1 is still a good way to keep diversity in the pipeline.
Got you. Okay, I'll find out.
Thank you for that. And then just a follow-up question for you, Louise. There was a question posted in the chat. How's your BNAB interferon trial going? Did COVID cause any delays?
That was a plant? No, that's funny. No. It's like, but in all seriousness, it's going very well. At first, we were very scared that the COVID was going to basically shut it down, but we've been able to recruit since June, and we hope to make full recruitment by the summer. And so far, things are going very well. There's a whole period of 6 months that individuals are taking both interferon and BNAbs, and the expectation from Marina's BNAb studies from Rockefeller is usually around 60% or 40 to 60% suppression that could be maintained. And now those studies did not select people for sensitivity for the antibodies, right? They basically were all comers coming in. We were one of the first studies to actually select sensitivity at the beginning, meaning that we knew that the individuals were carrying reservoirs that were sensitive to the antibodies that were going to be given. And up to now, I think of everyone that's been recruited, pretty much everyone has remained suppressed. So, which it's become now a question of, is it the addition of the interferon, or was it the fact that they were all sensitive to begin with? And so we're now recruiting the BNAb only arm in order to answer that question. Now, all of them will have an open-ended interruption coming up. So we're still going to be able to ask the question, did this make any difference? Or this was just a replacement of art? Or did we actually go beyond that? And but I think even if it's a replacement of art, it's still going to be kind of cool, because it will be the first study that ties the sensitivity to the outcome of suppression. If we are lucky and there's an impact beyond that, then obviously that would be awesome, but we'll have to wait. Now, this study, as you know, also introduced participant experience questionnaires at the beginning and end, that Kareem Dubé has been involved, and Chris and others as well from our CAB. And they also includes a viral load, at-home viral load study as well, that's being tested in this particular platform. So we're basically getting 3 studies for the price of 1, if you like, because we're getting the social science agenda, the home viral load testing, and the actual strategy itself, both in the context of art, which is one answer, or in the context of an open-ended interruption, which is a second answer. So I think we'll be able to get hopefully some useful information out of this.
Well, and I guess in maybe 3 or 4 years from now, you're going to rule the conference setting, because you're going to have all these studies releasing their data. And
I wouldn't go that far, but let's just hope that we have a poster or 2.
Yeah, so I wanted to kind of sculpt us towards an ending here. I wanted to hear from the researchers, how best might community members support cure research in the next 5 years. And then I'm going to ask the community here, I want to ask, what do you guys need from the researchers to help you do work in cure? what is it that you're going to need from them? So it's kind of taking both of those. So how can we, the community, help the researchers, and then community, what do you need from researchers?
Lots of questions.
Yes.
I will just say very briefly, I think it was kind of already said, but and it's been very evident in the chat, as we've been going through, but to keep demanding a cure and putting the pressure on us and the pressure on the, us scientists, and the pressure on the NIH to fund it.
Thank you. Next.
I would suggest, I think there's really, I hope there's going to be a groundswell of enthusiasm for virology and viral immunology amongst young people that is instigated, of course, by the COVID pandemic. And I hope that in part of that, we can attract new talent and new young minds to the HIV field as well, to continue to contribute to HIV cure. And one thing that I've seen is that for people who are junior in the lab, they really get excited when they have a chance to meet with the community and share their research with the people who are directly affected by these things. So I think the more opportunities we have to nurture and foster that enthusiasm, the better we'll set the stage for the future in our field, because funding, of course, is always a limitation. One thing that I find a limitation more and more is, the ability to recruit postdocs in the lab. And so I think we do need to make sure we continue to have that pipeline being refreshed.
Thank you. We want to take care to include diversity considerations in that as well. You talk about the pipeline, right?
Absolutely.
Okie doke. Others want to weigh in? We have lots going on in the chat box we'd like to discuss as well.
I think both Austria and Sweden because
how about we can get Linda and then Mark Milano to ask their questions. So Linda, why don't you go first?
Yeah, I think it would be really helpful if the docs would share and discuss their protocols and informed consent forms with their community advisory boards. And I imagine that most of you probably already do that. But I think it's, we do that extensively with DARE. We spend a lot of time, playing with informed consents, talking about inclusion, exclusion criteria and just toxicity issues and stuff like that. And as we do that, it really helps inform community advisory board members who can in turn take it back to their communities. So it's very helpful in, I think, not only us learning and being able to, send that, give that learning to other people, but also for the PIs to have a better, more patient-friendly protocol. So I think that's really important. And I love doing this. I think, it's one thing to hear a presentation if you're a community member. It's another thing to be able to ask questions. It just is like a flower blooming. You get so much more information and you learn things that you just didn't know from just hearing a presentation. So thank you all. It's really very important.
Thank you. And Mark, did you want to ask your question?
Yeah, I'm curious to bounce something off. One of the most important things we can take from webinars like this is how to disseminate this information. So I'm an HIV educator, and I hear constantly that the cure exists and is being hidden, and Magic Johnson has been cured. We all hear that stuff. And I spend a lot of time explaining why that is not the case. And I also explain the big question is, why is there no cure 40 years later? And I just want to bounce off with you the way I explain the reservoir and see if this is the best way to do it, if you have other ideas on how to explain the reservoir to somebody who is coming at this cold. I explain what a resting memory CD4 cell is. I explained that 1 in a million has HIV. And I basically say the reason we can't get rid of that is because they are in hibernation and the drugs cannot get into a cell that is hibernating. So kick and kill, I say, is a way to wake up the cell to get the drug is in there. But is there another simple way anybody thinks of explaining the reservoir to somebody who is brand new to this, so I can explain that HIV is able to hide and why it's able to hide and why the drugs we have today cannot get into the reservoir. Is there another better way to explain that? Or is my idea of hibernating cells and the drugs can't get in there the best way to do that?
Mm-hmm.
Thank you for that, Mark.
Anyone want to respond? Mark, I think your explanation mirrors mine very closely, except I get less technical. I don't use science terms when I describe it. In fact, I try to do like a metaphorical thinking about it, and I've had a lot of success. I start off a lot of my talks this way, and I'm putting it in the chat for you, the link to it. I call it the flower lesson, where I just use a field of flowers as an example. And there's one flower that's white and all the others are purple, or, an example like that. And I talk about HIV being seen and not seen that way. So I do it in a less scientific way, but it's the same thing. You give them a little more science in your description. I totally don't even take it into the science realm. I take it into a field like that. And so if you look at the video in the link, you can see what I've just managed to do, but it mirrors what you do. And I think what you're doing is what you have to do, is sort of break it down simpler. So if there are people who are thrown off by resting memory cell or talk of cells, then maybe you want to think of another metaphorical way of describing what's happening that's simple, that people can understand with being evaded, whether it's flowers or something.
I'm committed to actually teaching the science, so I do use those terms and I explain those terms. But to the researchers, is my explanation right, and is it a good way to explain it, or do you think there are better ways to explain the reservoir?
I think the context of your explanation is good, because you're obviously elaborating on the analogy and then explaining what it all means. So I don't know that there is anything wrong with that, if that's in the context of your presentation. I think the same thing is I've used that if you see in the a bird trying to find worms in the grass, so or bugs, that if the birds are your immune system and they're trying to pick them out, then they need to find them, right? And if you have a field full of rocks and you don't know which rock you need to lift in order to see the worms underneath the rock, then the bird can't lift the rock. So you need to find some other way to kind of get that rock moved so that the bird can see it, right? So there's so many ways that you could elaborate on the concept of an immune system, access to the drug, access to seeing where the virus may be hiding. I've seen other analogies of a pool full of those balls that you see on the kids' places, those that you basically just jump in, and all the balls are blue, and then there's only one ball that's red, and that's the reservoir, and you have to dive in there to try to find it because you can't really see it from afar. So I guess my answer to you is, I don't know that there's a right or a wrong analogy, as long as you put it in the context and you explain it.
Thank you for that. And Corrine also dropped some gems in the chat box about reservoir metaphors. And then thank you, Beth, for that reference as well. She also referenced a slide from Durban with a single red ball in a pool full of pink balls. Thank you for that.
Can I just say that, Luis, we love you. Sound like an Irish poet with all of these analogies. It's great. It's very helpful. You're not Irish too, are you?
No, sorry, Puerto Rican.
Beth commented, the Puerto Rican poet. We love it.